So let's talk about molecular evidence as a piece of support for for evolution. And what we're talking about here really are coming down to utilizing three different molecules in our cells. And those molecules are your DNA, your RNA, and your amino acids. I'm going to shorthand this as AA. And remember, amino acids are the building blocks for your proteins. All three of these molecules have a particular sequence within their building blocks. So when we talked about DNA and RNA, remember they are made of those nucleotides, right? And those nucleotides have different nitrogenous bases on them, so A, T, C, G, and U. And so we can put them together kind of like the letters of the alphabet to make different sets of instructions. And your amino acids work the same way, right? We looked at those R groups, and there's 20 different R groups. So the way that I put those amino acids together makes a particular protein based on that sequence. Again, it's like using letters in an alphabet to make a bunch of words. So when we look at this in our cells, there's something that you need to understand, and that is the fact that these three molecules have a connection to each other. So that DNA, right, is sitting in your nucleus, okay? And that DNA never really leaves that nucleus, but ultimately that DNA is what gets passed from parent to offspring, and it is what tells your cells what to do. It is your set of instructions. But if that DNA never leaves the nucleus, how do those sets of instructions actually get into the rest of your cell to tell your cell what to do? And that's where this idea of central dogma or flow of information molecularly exists in your cells. And we'll come back to this later this year and talk in more detail. But for now, understand that that DNA in your nucleus never leaves. So what we do is we make copies of just pieces of that DNA into RNA, okay? And so we copy the sets of instructions from DNA into RNA. That RNA then can then go leave the nucleus and go out into your cytoplasm, which is everything outside your nucleus. It's where your mitochondria are, your Golgi are, your ribosomes, so on and so forth. And those RNA molecules will then go specifically to your ribosomes. And your ribosomes are your protein factories. Okay. And so what does that mean? That means I'm going to take that copy of DNA that I made into RNA, and then I'm going to go to those ribosomes, and those ribosomes are going to read that copy of information to make the amino acid sequence, aka make the protein. Okay, so all three of these molecules are connected. DNA has the initial set of instructions for every protein your body could ever possibly create. The RNA then is just the copy for that particular set of characteristics I want to create that day, and then we turn that into the protein, which is the actual functional trait, right? So when we talked about those terms, genotype, right, which we've been shorthanding something like this, versus phenotype, which might be brown eyes, all right, the connection here is this is what's coded for in your DNA. That's the actual nucleotide sequence, all right, and you would have a certain sequence for the dominant trait, a certain different sequence for the recessive. And then when we put those sets of pieces of information together, they ultimately create your phenotype, which is the physical expression of that, which is the protein itself. So if I had blue eyes, that means my body makes a different set of proteins because I have a different set of DNA. And hopefully that makes sense. So what we can look at then for molecular evidence are those changes in DNA, right? Which will ultimately cause changes in your RNA, which will ultimately cause changes in your proteins. So if I take a look at those changes, aka those mutations, right? These are changes in DNA, which will cause changes in your RNA, which will cause changes potentially in your amino acid sequence, I can then look at how those species evolve over time based on the changes to a particular trait that they share. So that's what we're looking at over here. So here is a bunch of different species. They all contain this hemoglobin protein, right? Remember, polypeptide is another word for protein, right? Okay. And so we're looking at the percentage of amino acids that they share in common. So if I'm comparing, and this one is a human hemoglobin, human to human, that should be 100%, right? Because we're taking the same protein out of the same species. But if I compare that hemoglobin protein, which is also important to monkeys to function, we share 95% of those amino acids in common. If I compare human hemoglobin to a mouse, 87% of those amino acid sequences are identical. Go all the way down to human versus a lamprey's hemoglobin, and now we only share 14%. And so what this tells us is that over time, we know mutations accumulate. We know that there's sort of a background mutation rate that occurs. And so we can use this kind of like a molecular clock and be able to look at how many changes have occurred over time. The more differences that have accumulated, the more time that has